Okay, good afternoon, everyone. I'm Professor Laura Bishop, the Pro-Vice Chancellor for the Faculty of Science. It is my honor and pleasure to welcome you here today for the inaugural professorial lecture of Professor Claudia Mettke-Hofmann. It's been my privilege to be Claudia's colleague since she arrived at LJMU in 2007 to take up the post of senior lecturer in animal behavior. Even at that point, she already had an impressive academic pedigree, having degrees and a postdoc at the Free University of Berlin and postdoctoral fellowships at the Max Planck Center for Ornithology and the Smithsonian Institution. She lived up to all of our expectations. At LJMU, she was conferred a readership in 2014, and then, taking one for the team, became a subject leader in 2019. Throughout all this, she has been an excellent colleague and leader, all while delivering the best in teaching and research. Her research field is the evolution of cognitive behavior in birds, specifically how behavior and ecology interact. She's done significant research in the personalities of birds, but her research interests have not been restricted to birds. She's also published work on equids, also known as horses, and cockroaches. I am probably not the only person in this room to be mildly disturbed to learn that cockroaches have personalities. But let's just leave that there. I'm sure you, like me, are looking forward to hearing all about Claudia's research and her professional journey to the award of her professorship in 2022. Her contributions to LJMU and to the wider world of research have been truly remarkable. Liverpool John Moores University is marking its 200th anniversary of its founding as the Liverpool Mechanics and Apprentices Library in 1823. This lecture is part of our celebration of our bicentenary. Celebration is the operative word here, because we come together to mark Claudia's achievement and to learn and celebrate with her. I'm delighted to see so many of her friends, families, and colleagues from the School of Biological and Environmental Sciences and from beyond here this afternoon. I'm sure that you will all join me in giving a very warm welcome to Professor Claudia Mettke-Hofmann.
Thank you, Laura, for this really kind introduction. Can't be me. And you gave my talk already. So I just get this running here. Okay. So I have 45 minutes to summarize 33 years of research. That's less than 90 seconds for every year. So I think I better get going. And I would start, so life is weird and wonderful, and you never know where it will bring you. So I will start with some overview how I became a researcher and working in my field, and then I will talk about my research. So I was always interested in animals, how they behave, how they interact, how they learn, and one of my heroes was Konrad Lorenz, the founder of the animal behavior sciences. Here you see him with some goslings following him, demonstrating the imprinting process he discovered during his research. So the decision was easy: I will become a researcher, if it would be that easy. So I went to the job center and asked, what can I do with animals? Become an animal caretaker. But I thought that's too hands-on and possibly not so much observation. Then I can become a vet, but I can't kill animals. So this was the choice. And then you can study biology. And in Germany, this basically meant to become a scientist. So I enrolled at the Free University of Berlin and very soon joined the animal behavior team around Dietmar Todt. And everything I know about animal behavior, techniques, and the like, I learned in his group. There was only one caveat with him. He was interested in birds and birdsong, And I thought, that's really the most boring stuff you can think of. And then he had diversified into primates, primate communication. And I thought, that's a bit too close to humans. But at the end, for my final year dissertation, I settled on communication in Barbary macaques and collected my data under semi-free ranging conditions in southern France in Roque Amadou. While I was there, my parents came back, not while I was there, while I was analyzing my data with primates. My parents came back from a holiday and said, Claudia, we have a placement for you at the Max Planck Institute for Ornithology. You can stay there for 4 weeks and study birds. I thought, great, this is really the last thing I wanted to do. But what should you do? It was nice from my parents. So I went there, and basically on the very first day, the technicians took me into the woods, and we were catching birds. The first bird we captured was this beautiful chaffinch. And I couldn't believe it. This rusty red on the breast, the green on the back, the yellow on the wings. It was just the most beautiful bird I could imagine. And I was holding it in my hands. And from that day onwards, I was hooked on birds ever since, and I'm still interested in birds and love them. While I was doing my studies, I got more interested in play behavior, how animals learn. And when my placement supervisor, Bernd Leisler, said he has a PhD on information gathering and how this is linked to the ecology of a species, I said, that's exactly what I want to do. So the idea was, and this was something I was thinking about since a while, animals live in a variety of different habitats. Simple ones, complex ones, variable, stable ones. And the question was, do they all collect the same information? Or do they possibly pay attention to different parts of the environment, parts they are interested in that is relevant for them? And this was exactly the question of the PhD. So to give you some scientific background, intuitively you might expect that an animal should collect as much environmental information as possible, because the more you know, the better your decision making. But when you think a bit further, then there are costs of information gathering, like the time and energy that is spent on collecting information. Animals are exposed and they might be distracted from exploring, so they might be predated on. And the object or whatever they investigate might be dangerous. So there are quite a few costs to information gathering. And these costs have to be outweighed by the benefits. And this can be defined by the value of information. And the value of information is the difference in making a decision or a difference in making a decision with a payoff of an informed individual and an individual that is not informed. So basically, how much better is a decision of an individual that has information as compared to one that doesn't have information. And you can imagine that the value of information is higher in a variable environment where changes occur and it actually makes sense to explore, whereas in a stable environment where there are no changes, it's a kind of a waste of time. So my working hypothesis was that animals should not collect as much information as possible, but as much information as necessary. So I worked with 61 parrot species, covering all different types of groups like cockadoos, Amazons, macaws, budgerigars, parakeets, and lorikeets. And I collected these data at the bird park Walsrode. So the first thing I did, I went through the literature and went again through the literature and again to look for how they live in the wild, the habitats they live in, the diet they are feeding on. And I categorized 4 different habitats a simple open savanna habitat that is relatively stable, then open woodland that is a bit more complex but again quite stable. Then I had the forest edge, which is a very complex habitat and can be very variable. So animals can live more in the open part, they can live more in the edge, and then they can go into the interior. So there's lots of variation and change going on. And finally, I had the forest interior, where there are lots of strata to explore, so it's another complex environment. The next thing I did, I distinguished 10 different types of food, like seeds, fruits, nectar and pollen, flowers and buds, insects, roots, and the like, and calculated the proportion of each of these food types for each species. So how much they feed of each of this stuff. So the only question that was left, how can I measure information gathering? And it's quite difficult to identify whether an animal is looking around and is collecting information, or whether it's looking around looking for a mate. But it's quite easy to elicit information gathering by providing something novel. And then you get two different responses. One is neophilia, so neo is new, philia is being attracted to something, and so the animal would approach, explore, and collect information. On the other hand, novelty elicits neophobia, and phobia is avoidance. So this is the avoidance of novelty, and basically protects an animal from danger, because not everything that is novel has great resources and food or shelter, but can be dangerous, can be poisonous, a predator can be hidden there. And these two motivations are independent of each other, and depending which ones is more strong, it comes to approach, or it comes to avoidance, or they can be in conflict with each other, and then the animal approaches, or jumps back again, approaches again, and so on. So I was investigating both neophilia and neophobia, and here let's start with neophilia, the information gathering part. So I placed a novel object, a wooden ring, over a perch. So that's a neutral location where there is no food, no water nearby. So the animal isn't forced to approach, but when they approach, you would assume that they do this because they are interested in it and want to collect information. And then I measured the time to approach and correlated this time with the ecological variables I had extracted from the literature before. What I found was that species that live in very complex and variable environments, habitats like the forest edge, they were more interested in the novel object. They approached quicker as compared to those living in a very simple and stable environment like the savanna. And the reason for this is that this forest edge is very variable. There are many changes that occur, and animals have to keep track with these changes. So when you test them in captivity, they are also interested in any change in their environment. So they basically continue with their behavior they would usually show in the wild. I could also show that species that feed on difficult to find food like fruits and buds, they are very explorative, approach quickly, whereas species feeding on seeds that are plentiful available were very interested in the novel object. So what about the fruits? Imagine you have a tree with lots of fruits, and at one point this tree is depleted. So they have to explore the environment beforehand to find out where's the next fruiting tree that they can explore. And so they respond to changes, because any change in the environment, a change in the color, can indicate that there is a new fruiting tree emerging. So obviously not the tree, but the fruits that are newly emerging. And so they know where they have to go in the future. And so they respond to any changes in the environment. And finally, I could show that species that originated from islands were more explorative than closely related species from the mainland. So islands often have a lower predation pressure, so the costs of exploration are reduced. And also, there are often shortages over time at one part of the season because of the smallness of the islands, where the animals have difficulties to find food. So being explorative helps them to identify new food sources. So this was quite interesting and encouraging. And I next looked into neophobia, the fear of novelty. And here I placed a novel object next to the food. And what the response then is, the animals are motivated to feed. And then there's also the motivation to avoid the novel object. And when you measure the time to feed without the novel object, that's a normal feeding time, and you measure the time to feed with a novel object, then the difference between the two gives you a very good indication how fearful the individual is. So I first measured how do they respond just to the normal foods they put in, and on average, the species hesitated about 5 to 6 minutes to go to the food. The next experiment was I placed an identical but empty feeding dish next to the filled feeding dish and was really surprised about the responses. So here's a logarithmic scale, and you see the birds waited on average 20-30 minutes just by having an identical feeder next to it that was empty. So this was really, really interesting, and is really a warning to everyone who keeps animals. Don't forget anything next to the food, because animals might respond quite dramatically and might not go to the food for quite a while. And then finally, I placed a totally novel object next to the feeder, a cotton mop, you can see here, and most of the species really waited very, very long, 1.5 hours to 2 hours before they decided to go down and feed. So you can see that this fear response increases the more novel a situation is. And I could also show that when they are feeding on insects, they were particularly neophobic. So possibly living in a dangerous environment, because insects can sting, they can be poisonous. So species that feed on insects and encounter a more dangerous environment than others are more careful and stay away when there are any changes in their environment next to their normal food. So coming back to my initial question, do ecological conditions affect information gathering? Yes, definitely. And the time to approach varies with the variability in the environment, and the more variable their natural environment is, the earlier they explore. And when it comes to neophobia, again there was a link to the ecology. Species that live in more dangerous environments were more careful when there were any changes in their environment. So this was really, really exciting, and I really enjoyed my PhD. And I realized a bit later that there was a real application to it, a link to animal welfare. Because placing a novel object in a cage or an aviary is environmental enrichment. And my study basically showed that for which species environmental enrichment is particularly helpful. For those that come from a variable environment where there is lots of change and they are used to change, so we can enrich the environment by providing them environmental enrichment. Whereas there are other species that come from a very stable environment, they possibly don't appreciate to have this variable environment. So it helps people who keep animals to decide where to spend the money and where to enrich. I was quite excited about this research and wanted to stay with the parrots and wanted to follow a line where I had the impression during my PhD that there are some more differences in it. So I wanted to compare residents and nomads. So residents live in the same area year-round, possibly throughout their life, and you would expect that they have to collect information about this environment, any changes they're in, to be able to live there throughout seasonal changes. Whereas nomadic species, they leave the area when conditions deteriorate, so they wouldn't care about any environmental changes because they would move on. And this is something I wanted to investigate further. I wanted to change one thing. I wanted to have more controlled conditions because so far I had cages of different sizes, different numbers of species in, different numbers of individuals in. And this is not really how scientists work. There was no effect of it, but I still didn't like it. So I wanted to have my own cages, putting in one pair and testing them under controlled conditions. But how to do this? So during my PhD, most of the species I tested only once, because zoos only have each species presented once, and then in the next cage they have another species. But I had tested a few species several times in different zoos to show that the one response I got from the species is representative. But this was difficult going to ask different zoos to test one or two species. And then the same animal caretaker who had made me familiar with the chaffinch said, I know a bird breeder, he has exactly the bird species you need. I was a bit skeptical because I thought, does he really know the species I know? And also, wouldn't the cages be big enough? But we went there, and it seems the breeder was also a bit skeptical because he took annual leave to make sure that nothing happens to his birds. So I looked at the cages, they were really great, good size, and I started observation. And after a while, I realized there was another one observing, and this was a bird breeder observing me. So he wanted to find out what type of person is this? Can I trust her? Because obviously he had to go to work at one point again. Everything was fine. For the next couple of weeks, I was observing the bird there 6 days in a week. And it was really great because 10 o'clock I got a pretzel, 12 o'clock I got lunch, all from his mother. So it was paradise, all while I was observing the birds. So it was really good. And yeah, all went well. And the breeder was nice as well. Actually, he was really nice. He's my husband. So we are now married, I think, since more than 25 years or so. And he had a great idea. He said, why not borrowing the birds? You bring them for 3 weeks into your controlled conditions, do the experiments, we bring them back, it's cheap, and it's easy to do. Not so easy because we were driving thousands of miles. But yeah, we did it. And he was a really experienced bird breeder, very respected by the bird community, and he had the trust. Nobody would have given me a single bird, but he had the trust. So we got 80 pairs of parrots of 10 different species and tested them basically in the forest of the Max Planck Institute for 3 weeks each. So this was a great start. Working with breeders together and obviously working with Gerhard together. So I mentioned 10 different species. 4 of them were nomadic, so we had about half of them were lorikeets feeding on nectar and pollen, and the other half were broad-tailed parrots feeding on seeds, so also they have some variation. And we had 4 nomadic species, you can see here, 3 lorikeets and 1 parakeet. We had 4 resident species, 2 lorikeets, 2 parakeets, and then we had 2 species where you had to say they could be either or. They could be nomadic or resident, depending from where they were coming. But obviously, we didn't know where they were originally coming from. So another parakeet and another lorikeet. So I used my established testing protocol, where at this time I had 3 novel objects in their familiar environment. So they got used to this novel environment for a week, and I measured how fast they approach. And the idea was that the residents would approach quickly because they have to respond to any changes in their environment, to follow any seasonal changes in their natural environment. And this is basically what we found. The resident species approached very quickly, whereas the nomadic ones hesitated much longer to approach the novel object. So the residents paid attention to environmental change as we had expected. I then wanted to know, how do they respond to a novel environment? Basically, expecting that the nomads now would excel because they usually move away into a new area when conditions deteriorate. So I had the main aviary and opened 2 adjacent aviaries, left and right, which were furnished slightly differently, and we measured how long they spent in this novel environment, expecting that the nomads would explore for much longer. We were quite surprised about the responses because the nomads spent the shortest amount of time in the novel environment, whereas the residents spent much more time in this novel environment. So this was a bit unexpected, and I thought about what could be the reason, and it seems that these nomads, they spent only relatively short periods in each area. So for them, it doesn't make sense to collect a lot of information. They just find out where's food, where's shelter, and where's possibly where there's a predators. That's all what they need because they can't use long-term information because they might have moved on by then. Whereas the residents, when they go into a new environment, they do so to possibly find out, can they settle in here for the next year, for the next 2 years. So they need long-term information and thorough information about the long-term suitability of this habitat. And this explains why the residents spend more time in this novel environment than the nomads. So the nomads show superficial spatial exploration. So I wanted to stay, and the next one is this. This was kind of basically a new field of research because nobody had ever looked into differences in cognition between residents and nomads. And nobody knew how they collect information. And the results were really black and white, more or less. So I was really excited by this new finding and wanted to stay and wanted to follow this up. But I switched the nomads for migrants, and I also switched the parrots for songbirds. For the next couple of years, I studied primarily 2 species, the long-distance migratory garden warbler and the close related resident Sardinian warbler, and could show that they basically respond exactly the same like the resident nomadic system. So the migrants were not very interested in changes in their familiar environment, but they quickly entered a novel environment, but then only superficially explored this. Whereas the residents readily explored any changes in their familiar environment, but they hesitated to go into new environments, but then if they did so, they thoroughly explored it. So this was basically exactly the same what I had found with the resident and nomadic system, and showed that species that spend only limited amounts of time in a particular area, they don't invest a lot in information gathering because they can't use it in the long term. So this was quite exciting. But I could also show in the garden warblers, the migratory garden warblers, that they are highly neophobic. So when you place a novel object next to their food, they stay away for a very, very long time, whereas the resident Sardinian warblers, they relatively quickly approach and feed next to the novel object. And I could confirm this in my first field study, which I did at the Smithsonian Migratory Bird Center together with Russell Greenberg, in New World blackbirds, which you can see at the bottom. And the migratory birds, again, showed a very strong response when I placed novel objects around the feeding site, and they basically stayed away and rather moved into another area where there was no disturbance than feeding next to the objects. So they seem to be very susceptible to any changes in the environment, and this might be a problem with all the changes going on in our environment. And also, I had the opportunity to go into learning and memory and could show that the long-distance migratory garden warblers have a long-term memory for at least 12 months for a room that has contained food. And this helps them to remember high-quality stopover sites or remember the breeding site where they were coming from after a long time on the wintering ground. Whereas the resident Sardinian warblers, they have a short-term memory for only 2 weeks for exactly the same room, but they don't need a long-term memory because they live in the same area throughout their life and can update their knowledge whenever they want. So this research was really exciting and brought me more in the direction of conservation, because migratory species as a whole, they are strongly declining. And even though one of the main reasons might be habitat destruction, you can also clearly see that the mindset isn't in the right place. They are not really set up for changes in the environment because they don't explore them, they don't really respond to changes in their environment, they don't find out what is the advantage of a change. And they are highly susceptible to any change, so they rather go somewhere else which they know rather than exploring anything novel. So they are really repelled by any change in the environment. And the long-term memory might also be a hindrance because when they go somewhere where they have been before, the environment might have changed. So it's a dead end. So the migrants, they're not very well cognitively prepared to survive in our ever-changing environment. So for the remainder of the time, I would like to talk about the research I've done since I joined LJMU in 2007. I knew when I came here I couldn't keep 100 plus individuals of different species. So I needed a bird that was easy to keep, ideally a seed eater because they are cheap, and it needed to be a species I am obviously interested in. And the choice was the Golian Finch. They are seed eaters, they are relatively easy to keep, and they occur in different head colors. So I was able to continue with my comparative approach, not comparing information gathering between species, but comparing information gathering between different head colors. And I knew from earlier research that this is really, really exciting. There was also another line of research that had come up over the years, and this was individual differences. And I wanted to dive into this a bit more and basically combine both. So what are individual differences? Individuals differ consistently in their responses to environmental challenges. So you might have an individual that's very interested in these newly approaching or occurring leaves, whereas another one is more afraid of it. And interestingly, this response is consistent over time. So this individual, when confronted with another novel object or item, will again approach quickly, whereas the other one, again, will be more fearful. So that's kind of a trademark of this individual to respond to environmental change. And research had also shown that behaviors you wouldn't expect to be correlated are correlated with each other, like aggression and risk-taking. So I wanted to incorporate this into the birds with different color morphs. actually this is called individual differences personality, and we all know what personality is. We are all different. It actually comes from human research. Some of us are curious, others are more fearful, and this is exactly what the same you find in animals. So with the color morphs, there are quite a few species that occur in different color morphs: fish, reptiles, mammals, birds. And it was known that they differ in their behavior: aggression, courtship behavior, and predator avoidance, possibly linked to their conspicuousness, because an animal that is very conspicuous might experience more predation. And I wanted to combine the personality with research on color morphs. And the idea was that each morph, to survive in the wild, would need a specific combination of behaviors to persist. So to make this a bit clearer, imagine you have a conspicuous morph, here in red, might experience more predation, so it shouldn't be very explorative to reduce the risk of predation, so better stay behind. But then obviously it would miss out on opportunities, so it would need aggression to get its share and chase away the other individuals. So they would end up with this combination of behaviors. When you then have a color morph that is less conspicuous, so they can easily approach anything novel and exploit new resources. But they might be less aggressive because then they can do so in bigger numbers because there is safety in numbers. And when an individual combines these two behaviors, then they would have a good chance to survive. Whereas when you have an individual, for example, like this one here, the conspicuous one, it is not very explorative, that's fine, but it's also not aggressive. So it would starve because it wouldn't get access to food. Whereas this one up here, it is aggressive, but it's also very explorative, so it would possibly be eaten because it's discovered by a predator. So all these combinations, these birds would disappear, whereas only the individuals that have the right combination of behaviors would persist. And this is what I wanted to investigate in the Golian Finch. So the Golian Finch occurs in 30% red-headed birds, 70% black-headed birds, and less than 1% yellow-headed birds in the wild, and usually even in the same population. So when you go out there and have the luck to see them, you will see approximately these proportions of red heads and black heads. What is great with the Golian finches is that both sexes have this color polymorphism. So here you see the males with a very colorful deep purple breast. The females look the same, unbelievable colorful, but their breastband is paler. But you see they have red heads, or they have black heads. I don't have a picture of a yellow head, but both sexes have this color polymorphism, which is quite rare. So in most species, only the males have the color polymorphism. So this was a great system to work with. And I was lucky enough to have a PhD student early on, Leah Williams, and she wanted to investigate personality in the Golian finches. So she wanted to investigate 5 different behaviors that are usually associated with personality. So neophilia, the novel object at a neutral location, neophobia, a novel object next to food, risk-taking, they were allowed to forage for a few seconds on the feeder. Then we raised a predator silhouette and lowered it again. And then we measured how long does it take until the birds return to the feeder, knowing that there is a risk. We looked for social behavior, having a bird each in a cage and measuring how much time do they spend together, who initiates the connection, who breaks it up. And finally, we measured aggression, having a small feeder in the corner where only one bird could feed, and who starts the attacks first. So we first looked for consistency, because this is an absolute requirement for personalities, and tested this in neophilia, having the novel object on a perch, and measured this the first time here on the x-axis. And then a couple of weeks later, we repeated the experiment with a slightly differently colored thread. And this you can see on the y-axis. And you can see that the behavior within an individual, so each data point is an individual, was quite consistent. An individual that touched the novel object quickly the first day also did so the second day. And an individual that hesitated long the first day also did so the second day. So neophilia, was consistent over time. Neophobia wasn't, because the second object was too similar, so we basically couldn't get any neophobic reaction, but later on we could show that this is a highly consistent behavior. Risk-taking was consistent over time, social behavior wasn't, and aggression was consistent over time. So this was great. We had 3 behaviors with consistency, which is a milestone for personality studies. Then we looked whether any behaviors were correlated with each other and possibly formed a behavioral syndrome. And we could show that neophilia, the novel object on a perch, and risk-taking, returning to the food after a threat, was positively correlated. So an individual that quickly approached the novel object also quickly returned to the feeder after the threat. So this was great, because the Golian finches had personalities. The final question now was, are the differences in behavior linked to the different head colors? So we tested this, and for neophilia, this was the case, because the red-headed birds hesitated much longer to approach the novel object than the black-headed birds. And this is what we expected, because the red-headed birds are more conspicuous, so they should stay away and wait what the black-headed birds find out. We found the same result for risk-taking. Again, the red-headed birds waited longer and hesitated longer to come back to the feeder than the black-headed birds. And again, we expected this, because this is a really risky situation. And finally, for aggression, we could show that the red-headed birds were more aggressive than the black-headed birds, and this allowed them to stay behind. Let's see what the black-headed birds find out. Is it dangerous or not? And then use the aggression basically to chase away the black-headed birds. So this allowed them to get their share of the resources. So this was a really exciting finding, because the Golian finches basically signal to everyone else their personality. So the black-headed birds tell everyone they're neophilic, they take risk, but they are not aggressive. Whereas the red-headed birds tell the others they are non-neophilic, they are risk-averse, but they are aggressive. And this was a really exciting finding, and the very first for distinct color morphs. So this was a very early emerging field, and it was a really interesting result. So I continued with this research and was lucky enough to have an MPhil student from our own undergraduates, Georgie Eccles, who is now a PhD student in Texas. And she said, I'm really interested in the Golian finches, but I'm really interested in conservation. I want to have a twist in this direction. So this wasn't difficult, because the Golian finch is highly threatened, or is threatened in the wild. It's nomadic, it's a habitat specialist, and with all this habitat destruction and habitat loss, you really ask yourself, how do they survive in the wild? They have to move around, and they will for sure encounter many new and changed environments. And as a habitat specialist, how do they cope with this? Is this a problem for their decline? So we started to investigate how do they respond to a novel environment. And we created a cage which they could enter on their own that was representing more an open habitat. This is what they usually would prefer in the wild. And we had another habitat that was much denser and represented a habitat that they possibly wouldn't like. And we tested them in different head color combinations, pure red heads, pure black heads, and mixed head colors, to see whether they affect each other. And we found that both head colors entered the novel as an open environment faster than the closed one, basically confirming their open habitat preference. But then we got an interesting finding, because with a dense habitat, the red-headed birds entered the dense habitat faster than the black-headed birds. And this was really surprising, because we had expected the black-headed birds are less conspicuous, they are more explorative, so they would be the first one to go in. But we got exactly the opposite. We'll come back to this in a second. We also wanted to know how do they respond to a novel food? Because when you are nomadic, you will also encounter novel food. And how do they respond to it? Again, they were highly specialized in their diet. So we took their normal food, the seed mixture, and dyed it either green or red. And then presented the two feeders next to each other, one with a normal food and one with a dyed food, and measured how many visits do they make to the two feeders and also feeding. And we did this over a 5-day period to see whether they get used to the food. So on day 1, as you can see, both head colors clearly visited the familiar food more often than the novel food. And this wasn't surprising, because it was a really new experience to them. After 5 days, for the black-headed birds, the situation was basically still the same. They went for the familiar food, and they didn't really go for the novel food. But for the red-headed birds, they had changed their behavior, because they now went basically the same amount of time to the novel food as to the familiar food. So they had accepted the novel food into their diet, which was quite different to the black-headed birds. And again, obviously, it was very surprising. So what was going on here? So first I thought, did we possibly code the head colors wrong? That we just swapped the black heads and the red heads, but everything was okay? And then it suddenly dawned to me, I knew this pattern already. I had tested species before that were interested to investigate any changes in their environment readily, but then hesitated to enter a novel environment. And this was what I had found before in resident species. So the black-headed birds basically had a mind of a resident, whereas the red-headed birds, they behaved more like a migrant, not being very interested in changes in their familiar environment, but then readily going into a novel environment. And what about the food? It fits perfectly, because the redheads, they go readily into a novel environment. That's when they find novel food, and so they were more likely to accept this novel food into their diet than the black-headed birds. So it suddenly all made sense, and I was absolutely thrilled that finally the circle had closed from comparing residents and migrants on the species level, coming down to comparisons within species, comparing different head color morphs, and possibly finding a distinction between head colors and how they behave. One is more resident, and the other one is more a migrant or a nomad in this case. And they might help each other. So the resident might perform better when they are breeding and there are any changes, so they can help the redheads to get access to resources. Whereas when it comes to being nomadic after the breeding season, the migrant may take the lead and may help the blackheads to follow. And so this can help how this species can maintain this color polymorphism over longer periods of time. So it's a really interesting area of research. I'm still following this up, looking whether there is a kind of a behavioral syndrome combining different types of responses linked to residency and migratoriness, and following this up. But I would like to finish by giving the word, so to speak, to my birds, because I have a placement student in here, over there. She spent half a year with me from France, and we did some experiments where we had to cover the feeder, and the birds had to learn to remove the cover. And here you see the feeder, here you see the cover, and let's see how the birds behave. Off it goes, and having access to the food. So the next one is coming, removing the cover. And I'm pretty sure you believe me that these are the most extraordinary birds you can imagine, and that it's absolutely fun to work with them. So with this, I would like to finish and would like to thank you, would like to thank my team and all my colleagues for their support. Thank you.
Oh, thank you so much, Claudia. That was absolutely brilliant. Now, it is traditional at these events to give the speaker not only our acclaim and congratulations, but also a small token. So on behalf of the university and the faculty, I would like to present you with this wonderful gift.
Wow, thank you.
Thank you so much for this talk. I think we've all learned a tremendous amount about birds. No extrapolation to human behavior or personality is allowed whatsoever, okay? None, but thank you. It's been brilliant, and congratulations.
Thank you very much. Thank you all for coming. It's really a pleasure to work here, and it's really great to share this moment with all of you. And I hope you enjoyed the talk, and when you have any questions, don't hesitate to ask. Maybe not now, I don't know how much time we have.
I think most questions would be better with some refreshment, don't you all? So at this point, we'll invite you all to join us in the foyer for a little refreshment, and to ask any questions of Claudia that you might have. And thank you all.
Thank you.